Aphid forms. It is generally accepted that ancestors of Sternorhynchia aphids, adelgids, phylloxerids, etc. were like other hemiptera. They reproduced via eggs, Agawa and Miura, 2014, following sexual reproduction, Hales et al. 1997. However, whilst it is tempting to assume all adults were winged, many hemiptera exhibit brachypterous, macropterous forms. E.g. see here. Whilst remaining egg-laying, the Sternorhynchia evolved to alternate sexual reproduction with one or more generations of asexual reproduction equals cyclical parthenogenesis. Polymorph. From the Greek. Polymorphos. Polou equals many plus morphe equals form. Polymorphism. The coexistence, in the same locality, of two or more distinct forms. Independent of sex, not connected by intermediate gradations, but produced from common parents. Wiktionary, Mar. 2016. Modern aphids equals aphidity probably began as temperate tree-dwelling species, then diversified along with flowering plants, von Dolan, 2000. Yet, as Blackman found, half their species still live on trees for at least part of their life cycle, as do the other modern Sternorhynchia, Phylloxeridae and Adelgidae. Unlike other Sternorhynchia, aphids only lay eggs following sexual reproduction, usually in autumn, and are viviparously parthenogenetic for the rest of the time. Aphids now have far more species than phylloxerids and adelgids, and some aphid species have many body forms, others very few. Consider these four examples. 1. Neomysis circumflexus has never been found to have sexual forms, its primary host is lost, but reproduces parthenogenetically via viviparous females equals viviparae. Their nymphs mature into just two adult forms, winged or wingless, either of which can produce winged or wingless offspring. 2. Drepanocyphum platanoides is a single host species, which reproduces by cyclical parthenogenesis. For most of the year all the adults are green-winged viviparae, but in late spring nymphs mature into very pale, winged, viviparae that diapause during summer, then produce normal winged viviparae. In autumn, viviparae produce nymphs that mature to sexual forms, either winged sexual males, or wingless egg-laying sexual females known as oviparae. New and expanded pages. Delphiniobia machinite. Consider these four examples. 1. Neomysis circumflexus has never been found to have sexual forms, its primary host is lost, but reproduces parthenogenetically via viviparous females equals viviparae. Their nymphs mature into just two adult forms, winged or wingless, either of which can produce winged or wingless offspring. 2. Drepanocyphum platanoides is a single host species, which reproduces by cyclical parthenogenesis. For most of the year all the adults are green-winged viviparae, but in late spring nymphs mature into very pale, winged, viviparae that diapause during summer, then produce normal winged viviparae. In autumn, viviparae produce nymphs that mature to sexual forms, either winged sexual males, or wingless egg-laying sexual females known as oviparae. Aphid hossilist. Aldera. Viviparae maturing on the secondary host or wingless below. On their primary host nymphs, hatched from sexual. 3. Rhopalocyphum patti alternates host seasonally. On their primary host nymphs, hatched from sexually produced eggs, mature to distinctive wingless fundatrices equals founding mothers, one shown with nymphs below. Their daughters and granddaughters are also wingless, but covered with gray mealy wax, see below, and produce leaf roll galls. Crowding and poor nutrition eventually causes their gall-dwelling offspring to mature into greenish winged forms below, which fly to their secondary host. Viviparae maturing on the secondary host are wingless below, or winged, and are greenish with a rust-red suffusion around the base of each siphunculus. In autumn winged forms, known as gynoparae, not shown, return to the primary host, where their nymphs mature to wingless oviparae. Later in autumn viviparae on the secondary host switch to producing winged males, which fly to the primary host to mate with the oviparae, both shown below. Below are a fundatrix with her wax-covered nymphs, wax-covered adults and nymphs in their gall, a winged emigrant on the primary host, a colony on their secondary host, and a winged male with oviparae on the primary host. 4. Paracletus simissiformis host alternates, with a minimum 2-year cycle. Salazar, et al., 2015, on their primary host, nymphs, hatched from eggs, mature to distinctive gall-forming wingless fundatrices. These fundatrices produce leaf galls, and their nymphs become wingless viviparae specialized for gall dwelling. These viviparae reproduce themselves in the galls until autumn, then produce winged forms. These winged adults fly to their secondary host, where their nymphs mature to round green, wingless, root-feeding, ant-attended viviparae. Their nymphs either mature to round green wingless viviparae, or to flat white viviparae which feed on the hemolymph ophant larva. Their nymphs mature to flat white forms are, in autumn, to two different winged forms. One winged form gives rise to round green wingless root-feeding ant-attended viviparae. The other winged form flies to the primary host, and produces wingless sexual forms, males and oviparae. Some aphids, particularly host alternating species, are unusual in having many distinct forms like social insects, bees, wasps, ants, and termites have castes. A few gall-forming aphids are considered social, and have up to a dozen forms, including defensive, soldier forms. The multitude of possible aphid body forms has some important consequences. 1. They enable aphids to optimize for growth, reproduction, dispersal, estivation, symbiosis, host adaptation and defense. And to be optimized for one or more of those roles. Given which, the forms produced for any one species may vary seasonally. 2. Multiple body forms make life hard when trying to identify an aphid, especially for seasonal or seldom observed forms. 3. Owing to their rather unusual biology, the terminology for aphid forms is frankly rather a mess. Morphs, polymorphs and phenomorphs. Originally morph, just applied to visible traits, such as winged, wingless, or red-green, but this is no longer so. For example, some aphid species produce two outwardly indistinguishable winged viviparous forms. One form can fly, the other cannot. Conversely, winged aphids, upon reaching a new host, tend to optimize their reproductive capacity by autolysing their wing muscles. Whilst impairing their flying ability, it does not change their morph. A morph is the name given to specific groups of insects that are all of the same species but differ in appearance. The Amateur Entomologists Society Glossary, Feb. 2016. Unfortunately, morph, which you might assume to be equivalent to body form, as in morphology, has acquired so many usages and definitions as to be almost meaningless, and this confusion extends to its use as a suffix. One source of difficulty is that, whilst a few aphid body forms are dictated genetically equals genomorphs, most forms are dictated environmentally but not genetically equals phenomorphs. To explain. A phene is an individual genetically determined characteristic or trait which can be possessed by an organism, such as eye color, height, behavior, tooth shape or any other observable characteristic. Phenotype refers to a collection of phenes possessed by a particular organism. Wikipedia, Mar. 2016. All of the body forms we listed above, including males, but excluding funditrices, are produced by parthenogenetic viviparous reproduction. Aside from the occasional very rare mutation, every parthenogenetically produced female is genetically identical to her sisters, daughters and mother, which are therefore clones. Every female form with a clone, including the oviparae and funditrix, is therefore produced from an identical genetic blueprint equals genotype. Each body form evidently results, not from different genes, but from different sets of genes being switched on. The only genetic difference between a male aphid and his mother is she has two sex chromosomes, he has one, and brothers may vary accordingly. Thus Acyrthocyphon pisum males can be wingless or winged depending upon which sex chromosome they happen to inherit. Brandl et al. 2006. Some clones produce only winged males, some yield only wingless males, and some give both. Given so many body forms can be achieved by switching genes on or off, changing from one form to another sounds deceptively simple. Indeed, it is commonly but wrongly assumed that morph, in zoology, is shortened from metamorphosis, and implies plastic changes of form or appearance. However, unlike chameleons, once an aphid has matured into a particular form, it cannot change to another. Also, since intermediate forms tend to be suboptimal, form selection is generally an all-or-nothing choice. Intermediate forms are rare. For example, whether viviparae are winged or wingless is often determined by conditions such as crowding or nutrition, whilst an aphid is an embryo, or an early nymph. Occasionally those conditions change suddenly, causing adults to have intermediate forms such as the Brachypterus equals reduced wings, Simidobius oblongus. Below are a normal winged form, and a rare Brachypterus form next to a normal wingless form. Morph does not refer to atypical forms, local variants, malformed individuals, nor to a continuous variable such as body length, unless it has a strongly bimodal or polymodal distribution. Individual size commonly varies due to diet or maturity. Given the number of body forms potentially available within an aphid clone, much work has been invested in understanding what determines a particular body form, and how flexible that choice is. 1. Some body forms, it appears, must appear in a predetermined sequence. For example, a funditrix form, if such exists, must come first, then viviparous forms, and sexual forms last. One exception is Acyrthocyphon svalbardicum, which lives at very high latitudes, and commonly omits viviparous forms, Hales et al. 1997. 2. Trying to accelerate the transitions from funditrix to autumn migrants, or sexual forms, by for instance temperature changes, indicates aphids have a built-in interval timer. To prevent this, a minimum number of days, not generations, must elapse. 3. Many attempts have been made to produce adults normal to the primary host by moving viviparae from the secondary host ahead of the time when such flights usually occur, or to the secondary from primary. Such aphids generally feed poorly and die, but their progeny do not change morph. Indeed, it has been suggested that a crucial factor limiting the evolution of host-alternating aphids is their funditrices are highly specialized hence dependent upon, their primary host. Moran, 1988. 4. Many aphid species produce both winged and wingless viviparae. In some species either of these forms can give rise to either of the other. The stimulus to produce winged viviparae is often crowding, but in host-alternating species night length and temperature can be more important. 5. Some species produce viviparae in several distinct, alternative, color forms. These forms tend to produce offspring of the same color, and in some species these forms may also be winged or wingless. Whilst distinct color forms commonly arise within clones, presumably by switching on sets of genes, they can also be caused by bacteria, or by genetic differences. Acyrthocyphon pisum green and red forms, shown below, appear to be maintained by balanced selection from a predator Coccinella septempunctata, and a parasitoid, Aphidius urbi. When parasitism rates were higher than predation rates, red morphs were at an advantage. A number of biologists feel these latter two, number 4 and 5, alternate phenotypic sets of forms be known as phenomorphs or morphs, but genetically determined alternatives, such as winged and wingless males, be termed genetic polymorphs. Distinctive morphs are not limited to adult forms. Paraphilus acerus, for example, spends the summer months in a specialized estivating form, diaposing first instar nymphs. Below are normal 2/4 instar nymphs and estivating nymphs. Soldier forms, see below, produced by some gall-forming aphid species, are almost exclusively nymphs. Just one species is known to have an adult soldier form, Inba, 1988. A number of aphid species produce forms optimized, to some degree, for defense. For example, adults of Chaetophorus or Pterocoma species bear a large and specialized cotta, which holds their honeydew ready for ant collection. The ants, in turn, discourage birds and even browsing herbivores. Since all adults of those species are thus equipped, this is a monomorphic character. As Simon Leather describes, first instar nymphs of the gall-forming species Pemphigus batae fight each other for possession of an optimal galling site, whereas Epipemphigus nisimi fight for the gall. A number of Pemphigenae aphids and Hormiphididae aphids, tribes Pemphigeni, Eriosomatini, Fordini, and Hormiphididae, Cerataphidini, Nipponophidini, having formed a gall, produce nymphs optimized to defend it and or to repair damage. Pike and Foster, 2008. These are commonly first instar nymphs, but some use later instars. What proportion of nymphs are soldiers varies between species, but we found Hamamelis spetulinus appears, monomorphically, to produce partially sclerotized first instar nymphs with strengthened legs, shown below. Acknowledgements. We are grateful to the Amateur Entomologists Society for permission to reproduce an abstract from their website's glossary. Aphid. Aphids are small sap-sucking insects and members of the superfamily Aphidoidea. Common names include greenfly and blackfly, although individuals within a species can vary widely in color. The group includes the fluffy white woolly aphids. A typical life cycle involves flightless females giving live birth to female nymphs, who may also be already pregnant, an adaptation scientists call telescoping generations, without the involvement of males. Maturing rapidly, females breed profusely so that the number of these insects multiplies quickly. Winged females may develop later in the season, allowing the insects to colonize new plants. In temperate regions, a phase of sexual reproduction occurs in the autumn, with the insects often overwintering as eggs. The life cycle of some species involves an alternation between two species of host plants, for example between an annual crop and a woody plant. Some species feed on only one type of plant, while others are generalists, colonizing many plant groups. About 5,000 species of aphid have been described, all included in the family Aphidae. Around 400 of these are found on food and fiber crops, and many are serious pests of agriculture and forestry, as well as an annoyance for gardeners. So-called dairying ants have a mutualistic relationship with aphids, tending them for their honeydew and protecting them from predators. Aphids are among the most destructive insect pests on cultivated plants in temperate regions. In addition to weakening the plant by sucking sap, they act as vectors for plant viruses and disfigure ornamental plants with deposits of honeydew and the subsequent growth of sooty molds. Because of their ability to rapidly increase in numbers by asexual reproduction and telescopic development, they are a highly successful group of organisms from an ecological standpoint. Large-scale control of aphids is not easy. Insecticides do not always produce reliable results, because resistance to several classes of insecticide, and because aphids often feed on the undersides of leaves, and are thus shielded. On a small scale, water jets and soap sprays are quite effective. Natural enemies include predatory ladybugs, hoverfly larvae, parasitic wasps, aphid midge larvae, crab spiders, lacewing larvae, and entomopathogenic fungi. An integrated pest management strategy using biological pest control can work, but is difficult to achieve except in enclosed environments such as greenhouses. Distribution. Aphids are distributed worldwide, but are most common in temperate zones. In contrast to many taxa, aphid species diversity is much lower in the tropics than in the temperate zones. 2. They can migrate great distances, mainly through passive dispersal by winds. Winged aphids may also 2. They can migrate great distances, mainly through passive dispersal by winds. Winged aphids may also rise up in the day as high as 600 meters where they are transported by strong winds. 3. For example, the current lettuce aphid, Nasinovia ribisnegri, is believed to have spread from New Zealand to Tasmania around 2004 through easterly winds. 5. Aphids have also been spread by human transportation of infested plant materials, making some species nearly cosmopolitan in their distribution. 6. Forewing of the early Middle Triassic, early Onesian, aphid Vosegus triassicus, 7. An aphid fossilized in Baltic Amber, Eocene. Fossil history. Aphids, and the closely related adelgids and phylloxerans, probably evolved from a common aphids. Temporal range. Scientific classification domain, Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta order, Hemiptera suborder, Sternorhyncha infraorder, Aphidomorpha superfamily, Aphidoidea. Geoffroy, 1760. Ancestor some 280 million years ago, in the early Permian period. 8. They probably fed on plants like chordatals or Psychotophyta. With their soft bodies, aphids do not fossilize well, and the oldest known fossil is of the species Triosophis cubitus from the Triassic. 9. They do however sometimes get stuck in plant exudates which solidify into amber. In 1967, when Professor Olhai wrote his monograph Studies on Fossil Aphids, about 60 species have been described from the Triassic, Jurassic, Cretaceous and mostly the Tertiary periods, with Baltic amber contributing another 40 species. 10. The total number of species was small, but increased considerably with the appearance of the angiosperms. 160 million years ago, as this allowed aphids to specialize, the speciation of aphids going hand in hand with the diversification of flowering plants. The earliest aphids were probably polyphagous, with monophagy developing later. 11. It has been hypothesized that the ancestors of the Adelgidae lived on conifers while those of the Aphididae fed on the sap of Podocarpaceae or Araucariaceae that survived extinctions in the late Cretaceous. Organs like the cornicles did not appear until the Cretaceous period. 8.12. One study alternatively suggests that ancestral aphids may have lived on angiosperm bark and that feeding on leaves may be a derived trait. The Lochninae have long mouth parts that are suitable for living on bark, and it has been suggested that the mid-Cretaceous ancestor fed on the bark of angiosperm trees, switching to leaves of conifer hosts in the late Cretaceous. 13. The Phylloxeridae may well be the oldest family still extant, but their fossil record is limited to the lower Miocene Paleophyloxera. 14. Taxonomy. Late 20th century reclassification within the Hemiptera reduced the old taxon, Homoptera, to 2 suborders, Sternaryntia, aphids, whiteflies, scales, psyllids, etc. and Ochenaryntia, cicadas, leafhoppers, treehoppers, planthoppers, etc. with the suborder Heteroptera containing a large group of insects known as the true bugs. The infraorder Aphidomorpha within the Sternaryntia varies with circumscription, with several fossil groups being especially difficult to place, but includes the Adelgoidea, the Aphidoidea and the Phylloxeroidea. 15. Some authors use a single superfamily Aphidoidea within which the Phylloxeridae and Adelgidae are also included, while others have Aphidoidea with a sister superfamily Phylloxeroidea within which the Adelgidae and Phylloxeridae are placed. 16. Early 21st century reclassifications substantially rearranged the families within Aphidoidea. Some old families were reduced to subfamily rank, e.g., Ariosomatidae, and many old subfamilies were elevated to family rank. The most recent authoritative classifications have 3 superfamilies: Adelgoidea, Phylloxeroidea and Aphidoidea. The Aphidoidea includes a single large family Aphididae that includes all the approximately 5,002 extant species. 17. Phylogeny. External. Aphids, adelgids, and phylloxerids are very closely related within the suborder Sternaryntia, the plant-sucking bugs. They are either placed in the insect superfamily Aphidoidea, 18, or into the superfamily Phylloxeroidea, which contains the family Adelgidae and the family Phylloxeridae. 11. Like aphids, Phylloxera feed on the roots, leaves, and shoots of great plants, but unlike aphids, do not produce honeydew or cornicle secretions. 19. Phylloxera, Dactylosphaera vitifolii, are insects which caused the great French wine blight that devastated European viticulture in the 19th century. Similarly, adelgids or woolly conifer aphids also feed on plant phloem and are sometimes described as aphids, but are more properly classified as aphid-like insects, because they have no cotta or cornicles. 20. The treatment of the groups, especially concerning fossil groups, varies greatly due to difficulties in resolving relationships. Most modern treatments include the 3 superfamilies, the Adelgidea, the Aphidoidea, and the Phylloxeroidea within the infraorder Aphidomorpha, along with several fossil groups. 21. Sternaryntia. Psylloidea, jumping plant lice, etc. Alerodoidea, whiteflies. Coccoidea, scale insects. Aphidomorpha. Phylloxeroidea. Phylloxeridae, phylloxerans. Conifer aphids. Aphidoidea, aphididae, aphids. Internal. The phylogenetic tree, based on Papasotiropoulos 2013 and Kim 2011, with additions from Ortiz Ribas and Martinez Torres 2009, shows the internal phylogeny of the aphididae. 22, 23, 24. It has been suggested that the phylogeny of the aphid groups might be revealed by examining the phylogeny of their bacterial endosymbionts, especially the obligate endosymbiont Buchnera. The results depend on the assumption that the symbionts are strictly transmitted vertically through the generations. This assumption is well supported by the evidence, and several phylogenetic relationships have been suggested on the basis of endosymbiont studies. 25, 26, 27. Aphididae. Lock. Anatomy. Front view of wheat aphid, Schizaphus gramenum, showing the piercing-sucking mouthparts. Most aphids have soft bodies, which may be green, black, brown, pink, or almost colorless. Aphids have antennae with 2 short, broad basal segments and up to 4 slender terminal segments. They have a pair of compound eyes, with an ocular tubercle behind and above each eye, made up of 3 lenses called triomatidia. 11. They feed on sap using sucking mouthparts called stylets, enclosed in a sheath called a rostrum, which is formed from modifications of the mandible and maxilla of the insect mouthparts. 28. They have long, thin legs with 2 jointed, 2-clawed tarsi. The majority of aphids are wingless, but winged forms are produced at certain times of year in many species. Most aphids have a pair of cornicles, sipunculi, abdominal tubes on the dorsal surface of their fifth abdominal segment, through which they exude droplets of a quick hardening defensive fluid, 28 containing triacylglycerols, called cornicle wax. Other defensive compounds can also be produced by some species. 20. Aphids have a tail-like protrusion called a cotta above their rectal apertures. 11. 29. They have lost their Malpighian tubules. 30. When host plant quality becomes poor or conditions become crowded, some aphid species produce winged offspring, elates, that can disperse to other food sources. The mouth parts or eyes can be small or missing in some species and forms. 20. Diet. Woolly aphids on apple tree branch. Many aphid species are monophagous, that is, they feed on only one plant species. Others, like the green peach aphid, feed on hundreds of plant species across many families. About 10% of species feed on different plants at different times of the year. 31. A new host plant is chosen by a winged adult by using visual cues, followed by olfaction using the antennae. If the plant smells right, the next action is probing the surface upon landing. The stylus is inserted and saliva secreted, the sap is sampled, the xylem may be tasted and finally, the phloem is tested. Aphid saliva may inhibit phloem sealing mechanisms and has pectinases that ease penetration. 32. Non-host plants can be rejected at any stage of the probe, but the transfer of viruses occurs early in the investigation process, at the time of the introduction of the saliva, so non-host plants can become infected. 31. Aphids usually feed passively on sap of phloem vessels in plants, as do many of other hemipterans such as scale insects and cicadas. Once a phloem vessel is punctured, the sap, which is under pressure, is forced into the aphid's food canal. Occasionally, aphids also ingest xylem sap, which is a more dilute diet than phloem sap as the concentrations of sugars and amino acids are 1% of those in the phloem. 33-34. Xylem sap is under negative hydrostatic pressure and requires active sucking, suggesting an important role in aphid physiology. 35. As xylem sap ingestion has been observed following a dehydration period, aphids are thought to consume xylem sap to replenish their water balance, the consumption of the dilute sap of xylem permitting aphids to rehydrate. 36. However, recent data showed aphids consume more xylem sap than expected, and they notably do so when they are not dehydrated and when their fecundity decreases. This suggests aphids, and potentially, all the phloem sap feeding species of the order Hemiptera, consume xylem sap for reasons other than replenishing water balance. 37. Although aphids passively take in phloem sap, which is under pressure, they can also draw fluid at negative or atmospheric pressure using the sebareal pharyngeal pump mechanism present in their head. 38. Xylem sap consumption may be related to osmoregulation. 37. High osmotic pressure in the stomach, caused by high sucrose concentration, can lead to water transfer from the hemolymph to the stomach, thus resulting in hyperosmotic stress and eventually to the death of the insect. Aphids avoid this fate by osmoregulating through several processes. Sucrose concentration is directly reduced by assimilating sucrose toward metabolism and by synthesizing oligosaccharides from several sucrose molecules, thus reducing the solute concentration and consequently the osmotic pressure. 39-40. Oligosaccharides are then excreted through honeydew, explaining its high sugar concentrations, which can then be used by other animals such as ants. Furthermore, water is transferred from the hindgut, where osmotic pressure has already been reduced, to the stomach to dilute stomach content. 41. Eventually, aphids consume xylem sap to dilute the stomach osmotic pressure. 37. All these processes function synergetically, and enable aphids to feed on high sucrose concentration plant sap, as well as to adapt to varying sucrose concentrations. 37. Plant sap is an unbalanced diet for aphids, as it lacks essential amino acids, which aphids, like all animals, cannot synthesize, and possesses a high osmotic pressure due to its high sucrose concentration. 34-42. Essential amino acids are provided to aphids by bacterial endosymbionts, harbored in special cells, bacteriocytes. 43. These symbionts recycle glutamate, a metabolic waste of their host, into essential amino acids. 44-45. Carotenoids and photoheterotrophy. Some species of aphids have acquired the ability to synthesize red carotenoids by horizontal gene transfer from fungi. 46. They are the only animals other than two spotted spider mites and the oriental hornet with this capability. 47. Using their carotenoids, aphids may well be able to absorb solar energy and convert it to a form that their cells can use, ATP. This is the only known example of photoheterotrophy in animals. The carotene pigments in aphids form a layer close to the surface of the cuticle, ideally placed to absorb sunlight. The excited carotenoids seem to reduce NAD to NADH, which is oxidized in the mitochondria for energy. 48. Reproduction. Soybean aphid alternates between hosts and between asexual and sexual reproduction. 49. The simplest reproductive strategy is for an aphid to have a single host all year round. On this it may alternate between sexual and asexual generations, holocyclic, or alternatively, all young may be produced by parthenogenesis, eggs never being laid, and holocyclic. Some species can have both holocyclic and anholocyclic populations under different circumstances, but no known aphid species reproduce solely by sexual means. 50. The alternation of sexual and asexual generations may have evolved repeatedly. On this it may alternate between sexual and asexual generations, holocyclic, or alternatively, all young may be produced by parthenogenesis, eggs never being laid, and holocyclic. Some species can have both holocyclic and anholocyclic populations under different circumstances, but no known aphid species reproduce solely by sexual means. 50. The alternation of sexual and asexual generations may have evolved repeatedly. 51. However, aphid reproduction is often more complex than this and involves migration between different host plants. In about 10% of species, there is an alternation between woody primary hosts, on which the aphids overwinter, and herbaceous secondary host plants, where they reproduce abundantly in the summer. 2050. A few species can produce a soldier caste. Other species show extensive polyphenism under different environmental conditions, and some can control the sex ratio of their offspring depending on external factors. 52. When a typical sophisticated reproductive strategy is used, only females are present in the population at the beginning of the seasonal cycle, although a few species of aphids have been found to have both male and female sexes at this time. The overwintering eggs that hatch in the spring result in females, called fundatrices, stem mothers. Reproduction typically does not involve males, parthenogenesis, and results in a live birth viviparity. 53. The live young are produced by pseudoplacental viviparity, which is the development of eggs deficient in the yolk. The overwintering eggs. 52. When a typical sophisticated reproductive strategy is used, only females are present in the population at the beginning of the seasonal cycle, although a few species of aphids have been found to have both male and female sexes at this time. The overwintering eggs that hatch in the spring result in females, called fundatrices, stem mothers. Reproduction typically does not involve males, parthenogenesis, and results in a live birth viviparity. 53. The live young are produced by pseudoplacental viviparity, which is the development of eggs deficient in the yolk, the embryos fed by a tissue acting as a placenta. The young emerge from the mother soon after hatching. 54. Eggs are parthenogenetically produced without meiosis, 55, 53, and the offspring are clonal to their mother, so they are all female thelitoki. 1154. The embryos develop within the mother's ovarioles, which then give birth to live already hatched, first instar female nymphs. As the eggs begin to develop immediately after ovulation, an adult female can house developing female nymphs which already have parthenogenetically developing embryos inside them, i.e. they are born pregnant. This telescoping of generations enables aphids to increase in number with great rapidity. The offspring resemble their parent in every way except size. Thus, a female's diet can affect the body size and birth rate of more than 2 generations, daughters and granddaughters. 1156, 57. This process repeats itself throughout the summer, producing multiple generations that typically live 20 to 40 days. For example, some species of cabbage aphids, like Brevicorine brassicae, can produce up to 41 generations of females in a season. Thus, one female hatched in spring can theoretically produce billions of descendants, were they all to survive. 58. Aphid giving birth to live young. Populations are often entirely female. In autumn, aphids reproduce sexually and lay eggs. Environmental factors such as a change in photoperiod and temperature, or perhaps a lower food quantity or quality, causes females to parthenogenetically produce sexual females and males. 55. The males are genetically identical to their mothers except that, with the aphid's X0 sex determination system, they have one fewer sex chromosome. 55. These sexual aphids may lack wings or even mouthparts. 20. Sexual females and males mate, and females lay eggs that develop outside the mother. The eggs survive the winter and hatch into winged, alate, or wingless females the following spring. This occurs in, for example, the life cycle of the rose aphid, Macropsyphum rosae, which may be considered typical of the family. However, in warm environments, such as in the tropics or a greenhouse, aphids may go on reproducing asexually for many years. 28. Aphids reproducing asexually by parthenogenesis can have genetically identical winged and non-winged female progeny. Control is complex. Some aphids alternate during their life cycles between genetic control polymorphism and environmental control polyphenism of production of winged or wingless forms. 59. Winged progeny tend to be produced more abundantly under unfavorable or stressful conditions. Some species produce winged progeny in response to low food quality or quantity, e.g. when a host plant is starting to senesce. 60. The winged females migrate to start new colonies on a new host plant. For example, the apple aphid, Aphis pomi, after producing many generations of wingless females gives rise to winged forms that fly to other branches or trees of its typical food plant. 61. Aphids that are attacked by ladybugs, lacewings, parasitoid wasps, or other predators can change the dynamics of their progeny production. When aphids are attacked by these predators, alarm pheromones, in particular beta-farnesene, are released from the cornicles. These alarm pheromones cause several behavioral modifications that, depending on the aphid species, can include walking away and dropping off the host plant. Additionally, alarm pheromone perception can induce the aphids to produce winged progeny that can leave the host plant in search of a safer feeding site. 62. Viral infections, which can be extremely harmful to aphids, can also lead to the production of winged offspring. 63. For example, densivirus infection has a negative impact on rosy apple aphid, Disaphis plantaginea y_apple_aphid.htm reproduction, but contributes to the development of aphids with wings, which can transmit the virus more easily to new host plants. 64. Additionally, symbiotic bacteria that live inside of the aphids can also alter aphid reproductive strategies based on the exposure to environmental stressors. 65. The life stages of the green apple aphid, Aphis pomi. Drawing by Robert Evans Snodgrass, 1930. In the autumn, host alternating heteroecious aphid species produce a special winged generation that flies to different host plants for the sexual part of the life cycle. Flightless female and male sexual forms are produced and lay eggs. 66. Some species such as Aphis fabi, black bean aphid, Meadopolyphium dorhatum, rose grain aphid, Mysis persicae, peach potato aphid, and Rhopalosipum paddy, bird cherry oat aphid, are serious pests. They overwinter on primary hosts on trees or bushes. In summer, they migrate to their secondary host on a herbaceous plant, often a crop, then the gynoporae return to the tree in autumn. Another example is the soybean aphid, Aphis glycines. As fall approaches, the soybean plants begin to senesce from the bottom upwards. The aphids are forced upwards and start to produce winged forms, first females and later males, which fly off to the primary host, buckthorn. Here they mate and overwinter as eggs. 49.